Good morning. My name is Brett Pike. Today, we will discuss the basics of plant pathology. In particular, the difference between how a plant responds to pathogens in general and how it responds to specific tactics of a pathogen once that pathogen has developed an arsenal to, maybe arsenal is not the best word, perhaps a series of lock-picking tools that the fungi or the oomycete in my case have developed in order to sneak into the plant's leaf without setting off the alarm system. So let's start by describing how a plant leaf is put together. And there will be similar mechanisms at play in roots, stems, flowers, all parts of the plant. But the most surface area, and therefore the most biomass, is in the leaves. And so that's what we will talk about. That's where the largest part of the life history of these pathogens takes place. So this is a leaf in cross-section. This diameter is perhaps 0.2 of a millimeter. This is called the lamina, like a laminate. It's a flat leaf. You know, and leaves can come in various shapes and sizes, colors, but the basic structure is consistent from one to the next. And so I'm using green because plants are green because of a pigment called chlorophyll, which takes energy from the sun and uses it to split water from the roots and oxygen from the air to make energy. And it uses that energy to make carbohydrates. It's very interesting. It's actually the basis for the food chain. The vast majority of food starts out as plant-generated carbohydrate, which is then consumed first by herbivores and then by omnivores or carnivores, and so on. But it all begins with sunlight striking the surface of a leaf. So this is empty space. And these green cells, the ones on the top side of the leaf, are called palisade parenchyma. It's called a palisade, I think it's an architectural term, because they're stacked closely together, like bricks in a palisade. So that makes sense because if the sun is above you, it makes sense to have most of your light harvesting organs on the side which is closer to the sun. So then underneath you have spongy parenchyma, which is a looser network of similar cells surrounded by air. And there are pores in the underside of the leaf and also on the top side, but mostly on the underside, called stomata. I think stomata is the plural, and I think an individual one is called a stoma. And there's a couple on the top as well. And these stomata are able to open and close, and that allows oxygen, O2, or to enter the leaf to participate in photosynthesis, and water vapor or H2O, to diffuse out. And this is typically the limiting factor in how rapidly this overall reaction can take place. So these, I think these are individual cells. And these are chloroplasts, which do the actual reaction of taking energy from the sun and oxygen from the, CO2 from the air. This is not correct. It is CO2 that diffuses in and oxygen that diffuses out. And there's a significant difference. Oxygen and water vapor that diffuse out. The water comes into the roots and then is transported through the vasculature, the xylem. Perhaps, well, the phloem also is an aquatic solution, but most bulk water diffuses through the xylem. And photosynthesis takes place in these cells. So the net result of photosynthesis is that you get a concentration of sugar. These leaves tend to function as source tissues for the rest of the plant. The sugars are synthesized here and then exported to the growing tip, to the roots, to the flowers, and those are sink tissues that consume sugar. So the concentration of sugar is highest here so that the sugar flows down its concentration gradient to those other parts of the plant. So it's an attractive target for parasites, such as a variety of insects. Draw an aphid here, and we'll try to stick its stylet in, and I think they have 6 legs. And suck out carbohydrate from within the plant without the plant knowing it's there. Bacteria all over the place, covering this entire surface, good bacteria and bad bacteria. And also filamentous pathogens. There's 2 main types of those. Fungi, such as most filamentous pathogens, molds, mildews, other types of fungi, but molds and mildews are the 2 main types that you may be aware of. And oomycetes, which are different genetically, but have a similar life history. So I'll describe them both here together. And so the way that an oomycete, for example, Phytophthora is one that's, as the most species known. The one that we work on is called Bremia. Bremia lactucae is specific to lettuce, and they have a similar strategy for exploiting a leaf. One of the big differences between these 2 is that fungi are distributed via airborne spores that float through the air and then land on the surface of a leaf and then try to penetrate. Oomycetes tend to require surface water to be virulent. They are not light enough to float around. They have to swim. They have these structures called zoospores, which are biflagellate, which means they have 2 flagella. And the flagella are of different types. One is a tinsel type, and the other one is a whiplash type. And these, for a long time it was thought that these were types of fungi, outline types of fungi, but they're actually quite different. And the closer you look, the weirder they are. This arrangement is not found very commonly, but it appears to work well for the few oomycetes that are out there. So a zoospore, which is diploid, it has two copies of its genetic information in it. Let's say that we are talking about a stomata on the top side of a leaf, although this probably happens more frequently on the underside, For purposes of ease of illustration, I'll draw it on the top side. So say in the fall you have frequent rains, you have water falling from the sky to the ground. You have many spores, zoospores or fungal spores, resting in the soil, and they splash up from the soil onto the stem or onto one of the lower leaves of the plant. And because it is raining, there is a thin film of water on the surface. And so you have a zoospore swimming in that surface water. And so it finds a stoma. Now, typically, stoma can be either open or closed. That's regulated by turgor pressure. There's two kidney-shaped cells called guard cells that, when they are dry, they are closed, and the stomata does not admit water or air or, hopefully, pathogens. But when they swell with water, they swell up, and an aperture becomes available for entry. So the zoospore is able to locate these stomata and penetrate into the leaf and then germinate. And at that point, it begins growing what's called hyphae, which is like a root of a fungus. It's a long filament. That's why these are known as filamentous pathogens that can then penetrate throughout the plant and is able to extract nutrients along the way. And so the basic function of any biological system is to propagate itself. So once this oomycete has collected enough carbohydrates, proteins, etc., from the plant, it's able to locate another stoma, and this almost always is on the underside of the plant, I think. Well, that's not true. On both sides of the plant. This would perhaps be more accurate if it was going the other way. Then it everts a structure called a conidiophore. And this is not to scale. They're actually much smaller than this. But this conidiophore has at its tips spore-bearing structures that then make more spores that then are able to be discharged into the environment. And so when these become extremely dense, that's when the symptom of this disease becomes visible on the underside of the leaf or the underside of the leaf as a mass of gray fuzz, or in the case of powdery mildew, you may just have dry powder on the surface of the leaf. In the case of a downy mildew or a gray mold, it continues to be filamentous following eversion from the leaf. And then each one of these is then able to spread via wind currents, via water, or landing on an animal of some kind, and then is able to disperse throughout the system. So this is a highly virulent pathogen where once a single spore infects one plant, you can easily have millions or billions or perhaps trillions of spores that can then rapidly take out an entire field. You could lose an acre in a couple of days. So getting some measure of control of this process so that we can harvest the leaves, and lettuce, of course, the leaf is the harvest. If you're harvesting the fruit or the roots, then you have a different set of problems. But for lettuce, it's especially important that the leaves not show any signs of damage, of fungal damage, for reasons of consumer acceptance. Also, this weakening of the the epidermis of the plant can lead to secondary infections. So when you see that E. coli has been infected with, when lettuce has been infected with E. coli, that frequently can take place starting with a bremia infection that creates lesions that then E. coli is much more able to penetrate. E. coli on its own would have a much reduced ability to get access to a cell. Of course, if you're dealing with a chopped lettuce product that is making a slice across a leaf, then that makes it far easier for an E. coli on the surface to get into the inside and propagate itself to a level where it becomes problematic. So this is a very basic life history of the bremia pathogen. And in the next video, I will talk about what is happening at the interface between the filament and the cells that it's attempting to extract nutrients from. So thank you for watching.